Heligmosomoides polygyrus, or H. polygyrus, is a naturally occurring gastrointestinal helminth found in mice. Due to its close relation and resemblance to prevalent human helminth infections, H. polygyrus is used as a model of hookworm infections in humans. It has a direct life cycle within the host, as it initiates with the parasite being ingested in its infective stage as L3 larvae by fecal-oral transmission. When L3 larvae reach the duodenum, they invade the intestinal wall by penetrating and causing structural damage to the epithelial cells. The larvae then migrate to deeper layers of the duodenum, and more specifically to the subserosal layer, where they encyst and further develop into their L4 form. The L4 stage of the parasite is critical, as during the time it has burrowed into the tissue of the intestinal wall, it releases molecules that modulate the host immune response. One such immune response is the interleukin-33 or IL-33 pathway. IL-33 is a signaling molecule released upon epithelial damage. It is part of the type 2 immune response, and it plays a critical role in both fighting infections by helminths in the cutaneous or mucosal sites and in the pathophysiology of allergic diseases like asthma when released irregularly. This is because it coordinates and recruits the activation of cells which are responsible for the development of asthma. Such cells include T helper type 2 cells, type 2 innate lymphoid cells, and dendritic cells, amongst many others. To exert its function, IL-33 binds onto the ST2 receptor, which transmits signals to the intracellular space and is expressed on numerous cells. While in the L4 stage, H. polygyrus modulates the IL-33 response in multiple ways, all of which simultaneously halt the activity of the nuclear cytokine. For example, the parasite releases two proteins, with each having different effects on the IL-33 response. The HP-ARI protein halts IL-33 activity in two ways, by binding and blocking the cytokine directly, and by binding DNA and tethering IL-33 within the necrotic epithelial cells. The second protein, HP-BARI, binds onto the ST2 receptor, blocking IL-33 from attaching onto it and hence preventing it from activating further signaling pathways. Between 8 and 10 days after the initial infection, H. polygyrus returns back to the intestinal lumen in its adult form. There, it proceeds to mate and produce eggs for the next several weeks. By understanding how helminths like H. polygyrus are able to regulate the immune response of the host, new treatments for immune-mediated diseases, as well as new ways to combat such pathogens, which affect around a quarter of the world's population, can be developed.